So now moving on to different types of cell. The first cell that I have made here is a bacterial cell, which is actually a prokaryotic cell. This is a prokaryotic cell, which I have already told that it do not contain a proper nuclear membrane. The nucleus of a prokaryotic cell, which is a primitive nucleus, that means it lacks nuclear membrane. All the nuclear or chromatin content is lying naked in the cytoplasm of a cell. See, this is a DNA content, or this is a chromatin content, which is without a nuclear membrane, and this is a main characteristic feature of a prokaryotic cell. Whereas if you see a eukaryotic cell, which I have made here is an animal cell, you can easily differentiate that this nucleus or this chromatin material is surrounded by a complete envelope of nuclear membrane, whereas this nuclear membrane is absent in the prokaryotic cell. The various other features of this bacterial or this prokaryotic cells are that it contains a capsule. This capsule is actually formed of a mucilage layer. This is formed of a mucilage, and this mucilage is not normally present in every bacterial cell, but however in unfavorable conditions, so as to prevent this cell from drying. During those unfavorable conditions, this bacteria develops a mucilage covering around its membrane, and that's why we call it as a capsule, or it is a protective sheath. The main component of bacterial chromosome is that bacterial part is that it contains a chromatin material, which is known as bacterial chromosome, without a nuclear envelope. However, apart from this main bacterial chromosome, bacteria also has some extra chromosomal DNA, and that extra chromosomal DNA is known as a plasmid. This is extra chromosomal, which is present apart from the main chromosomal DNA content. The chromosomal DNA is present in the bacteria, but apart from this, it has some extra material, extra chromatin material, and that is, we call it a plasmid. The next part is that it contains mesosomes. This is a mesosome, and this is also a mesosome. This mesosome is involved in the respiration of the bacteria. The next component is that it consists of a semi-fluid structure. The semi-fluid part, where this all organelles are involved, this is known as cytoplasm. This cytoplasm is again present in the animal cell as well as in the plant cell. This is a fluid-like structure which is present throughout the cell, in which all these organelles are actually kept in. So this is known as cytoplasm, which is a semi-fluid structure. The next part is that glycogen molecules, which are also present in the bacterial, and flagellum is present in the inside the bacterial chromosome. This flagella helps in the movement of a bacteria cell. However, apart from flagella, sometimes one more protrusion is present in a bacterial cell, which is known as pili, or basically we call it sex pili, because this sex pili helps the bacteria in conjugation. It is involved in conjugation. So these are various parts of a bacterial chromosome or bacterial cell. Now moving on to next cell, which is animal cell. Animal cell, I have already told, it is a eukaryotic cell. That means it contains a true nucleus. This is a true nucleus. True nucleus means that it is surrounded by a prominent nuclear membrane, which was absent in the prokaryotic cell. This prominent nuclear membrane is surrounded by these ribosomes, which these ribosomes are kept on a organelle which is known as endoplasmic reticulum. Endoplasmic reticulum are classified into two categories. One is smooth and one is rough. Rough endoplasmic reticulum is meant by that endoplasmic reticulum which contains ribosomes on its surface. These red colored parts are of ribosomes, and these ribosomes are present on the endoplasmic reticulum. When endoplasmic reticulum contains ribosome, it is referred to as rough endoplasmic reticulum. But however, smooth endoplasmic reticulum is that endoplasmic reticulum which do not contain ribosomes, and this endoplasmic reticulum lies periphery of this nucleus. Nucleus also contains nucleolus and a DNA chromatin material inside it. The next important component of this animal cell is that Golgi apparatus. Golgi apparatus is involved in secretion in transporting the vesicles. So this is a Golgi apparatus, and these are the various vesicles that are removed from the, or that are protruded out of this Golgi part. So this part is this, actually this part is when it removes or it buds off from this whole Golgi apparatus, it is referred to as Golgi vesicle. We call it as Golgi vesicle because it is budded off from a complete Golgi apparatus. The next thing is vacuole, which can be a fat storage or can be a food storage vacuole. The next part is lysosomes. Another part that an animal cell contains are the secretory vesicles. Secretory vesicles are involved to secrete the material in and out of the cell. The another prominent part is that peroxisomes and mitochondria, which is a powerhouse of cell. So this powerhouse of cell is present in, this is present in eukaryotic cell, but it is absent in prokaryotic cell. So all the ATP which is required for energy, it is synthesized by this mitochondria. So these are various prominent features that are present in the eukaryotic cells, but they are actually absent in the prokaryotic cell.